All right. Hi, everyone. Thank you for joining us today. And this seminar is brought to you by nonprofit organization iExplore. iExplore is dedicated to providing an extensive online study and communication platform for students throughout the world. I am honored to introduce our seminar presenter today, Ryan Kern, who also happens to be our president of iExplore. He will be telling us all about biology, specifically natural selection and evolution. He will be guiding us on a journey through the discovery and application of evolution. So Ryan, take it away.
Thank you very much, Kathy. So like she said, let me grab my pointer here real quick. I'm Ryan Kern. I'm 10th grader at Troy High School. I really love playing water polo, as you can see in this picture here. I love playing the piano as well. And one of my main interests here is really just biology. I'm really passionate about it, which is why I'm teaching this seminar to you guys. And in the future, I would really like to be a doctor someday. So I hope you guys are excited as I am for this seminar. It's gonna be a really great one. So today we're going to be talking about evolution, which is really how we came to be. And we're not only going to talk about it in like our human context, but also just broader as well. And I hope you guys will have a good understanding by the end of the seminar. First, we're going to start off with the dictionary definition of what evolution is. So it says the process by blah, 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 like is thought to have developed and diversified. So you guys can go ahead and read this real quick. I think it's good just to have in the back of your mind. In reality, evolution is like this, but sometimes it's a little different. And if you look right here where I'm pointing, you see that one of the synonyms is natural selection, which we're going to do a comparison of natural selection versus evolution in later slides, as we'll see that they're actually very similar and deeply like connected. Yeah. So we've been talking about evolution, right? We found the definition, but who came up with this idea? Did it just appear out of nowhere? Like what happened? And so we're gonna ask you guys what you guys think. What do you guys think that it really actually came from, and who came up with the idea? So the first, here we have Mickey Mouse. Do you think Mickey Mouse came up with it? Or maybe Gregor Mendel, Albert Einstein, or Charles Darwin? So I don't know about you guys, but Mickey Mouse is looking like a pretty good option. We'll give everyone just a couple more seconds here. Hopefully this is not too hard of a question. Great job. Majority, as always getting the correct answer. Great job, Charles Darwin. Yep. And so here we have pictures of Charles Darwin. This is him when he was younger. This is him obviously older. So Charles Darwin was a very important figure in like really discovering and coming up with the idea of natural selection. And he really did this on a very interesting expedition as, sorry, there's my dog in the background, as he went on this journey on this ship called the Beagle. And the Beagle was going around the world and traveling, and he was really just like really traveling, but also like finding more about different like animals, because when you go throughout the world, there's so many different animals, species of plants, of whatever. And he was really just writing it down in his notebook. He was documenting, he was trying to draw them, and he was taking very good notes. And that's why like he learned a lot from this expedition, as well as we'll see, like to actually study evolution here. And so that leads us to our next question is, which animals did he actually use to study evolution and come up with this idea? Because he's been traveling all over the world. So which ones did he actually use? And I think we can share the poll now. Yeah, thank you. So A, finches, they're just a type of bird. B, blue whales. C, kangaroos. Or D, fish. So give everyone just like a couple more seconds. Yep, again, everyone keeps getting it right all the time. So proud of you guys. Finches is correct. Here we see, oh, these are the four options. Nope, it wasn't blue whale. Nope, it wasn't fish. No, it wasn't kangaroo. But yes, it was finches. So how did he discover evolution using these finches, right? Because like we can all see birds, but like, what did he actually do and what did he think about to come up with this idea of evolution? Well, there's these things that the islands he went to were called the Galapagos Islands. And on his journey on the Beagle, remember, is his ship, he went around, like I said, around like a very good portion of the world, trying to find and document new species and really learn more about them. And one of his stops was the Galapagos Islands. Here we have a picture of a finch that would be on this like kind of grass area here, or sorry, not grass, excuse me, like where there's forests and shrubs and trees. But if you look here, the Galapagos Islands are very small. There's not like, it's not like a mile across where there's just land everywhere. There's like water, you can see it on both sides, all like 3 sides. So they're not very big islands, which is why it's very interesting, because if you think about it, sometimes you'd be like, well, we want a really big island. You know, we want to have a ton of like animals and everything. But these islands were really good because they were decent sized. They weren't too tiny for like the population of, let's say, like animals and birds to be really affected by anything that happened, that like really immediately affected. And we'll talk about this later. But they weren't too big to the point where there's just like too many species and it's kind of hard to focus and really hone in on certain ones. So this was like basically the outdoor lab, if you will, for many of these people that were studying evolution. And this is why like Darwin was documenting it, and this is where he got his first ideas. Here, where I'm going to go over a couple of the main species on these islands. You see these huge lizards, and these are like one of the main species on this island here, and they just like to bask in the sun. They can actually swim in the ocean, and that's where they spend a good amount of time too. Here we have the famous giant tortoise here. These are very kind of rare, and they're very unique to this island. That's why with Galapagos Islands, if you're a visitor, they're very like strict with trying to keep your distance from the animals, because they want to preserve it as much as possible. As you can see, when we look back at these islands, there's not like buildings here, there's no hotels, there's no resorts. They're trying to keep it as pristine as possible and protect it. Here we have just an overview of like the finches, right? So these were like the different types of finches that he kind of saw. And there's actually going to be more like variations, but I just want to give you guys like an intro about really the main difference was kind of size, but also just the beak. The beak you can see right here on number 4, very small. 3 is like slightly bigger, 2 slightly bigger, and then 1 is very massive. We'll talk more about the specific functions of the beak and why it changed, or just why it changed based on their food supply. Oh, excuse me. And here we have a lot more of these finches that are actually on the Galapagos Island today. And you can see that there's many like variations here, and they're not just like all one uniform. So this originally was gonna be questions, but today we have a good amount of material. So I would like everyone to enjoy the picture real quick, but I wanna get through all the material. So if you guys have questions, please write it down and we'll get to it at the end. But I really just wanna get through the material because it's gonna be some fascinating stuff and I want you guys to learn it and hopefully also answer some of the questions you guys have as well. Oh, and a bonus as well, from the last seminar, we're actually going to answer some questions that we didn't get to in the end here too. So I want to make sure I get to that as well. Okay, so now we're going to talk about what the difference is between evolution and natural selection. Again, let's go to the textbook definition here, comparing and contrasting them. We already know what the definition for evolution is here. And now we see natural selection, but really evolution is more like natural selection, but like over a very long period of time. And we're going to go more into depth what natural selection is, but all you need to know is that really natural selection can cause like evolution, but evolution is not really just a very short-term sort of change that we would say is actually natural selection, is more the short-term word for it, and evolution would be seen as everything as a whole of different species as well. This is actually going to all rely on the mutations we talked about last seminar. If you remember, or if you weren't there, go to our YouTube channel to check it out. Here we have the mutations. Remember, it was a single nucleotide. If you don't know what that is, make sure you check out the DNA video. That changed it. This in turn changed the protein, and we remember how important proteins are, and this changed the shape of the sickle cell. This is very dangerous because if you have both of the alleles for this, which will be a future seminar that will be covered. It can actually block your blood flow here, and it's called sickle cell anemia. Just like how we said, we went over DNA replication and stuff like that, how when these proteins and the machinery of the cell, or the machinery we can say, is trying to go through this DNA relatively fast, and that's why sometimes it's very, very accurate, but sometimes there's going to be a change, there's going to be a mistake. And even though there are different layers that we talked about, there's like the different layers of proteins and other machinery that comes in specifically to check and change the base pairs out if there's a problem, sometimes it will get passed, and this is how those mutations occur. It's just like, if you were to think about it, you're working in a factory all day, and then your boss is like maybe just yelling at you to go faster and faster. Eventually, even though, let's say someone checks it, eventually if everyone's trying to go super fast, there's going to be some mistake. But compared to many other types of cells that we can talk about too, and like stuff like viruses as well, our DNA is actually very, very accurate for replicating. So that's a very good thing to know as well. Most of these mutations will end up not really helping or changing anything, or they will be like harmful, but some, again, a very, very small percentage again, are going to be beneficial. So this helpful mutation that is beneficial is actually going to give these species an advantage over other organisms. Now we're going to go into kind of an analogy, because I know I talked about this all kind of high level, right? But like, what does that actually mean when we think about just natural selection and like how genes are passed on, because we talked about some DNA as well. So here we're going to use an analogy of a race. Think of surviving as a race, and you have to run 5 miles to win the race. Pretty simple. Here we have good mutations, right? Let's say you have 2 full bottles of water, you have this really muscular body here, and you have perfect running shoes. So that's going to give you the best ability to actually run and win this race. However, if you don't have the good mutations, but you don't have the bad mutations either, you just have this smaller bottle of water, you have these not so muscular legs, but they're okay, and then you just have these like flip-flops to run in. However, if you have the bad mutations, you don't have any water and you're going barefoot. So we can kind of relate this, because if we look here at the next slide, the winner is going to be able to pass on their genes. So let's go back to the really muscular person. This person is going to have the more muscular and stronger genes, and it's going to be able to pass that on once it wins a race. And because it has these muscular genes, let's say for like a mutation that happened, right, that was good, it's going to be like a positive cycle here, because it has the best chance to win the race, since it has this really strong body, perfect like shoes and equipment and water, and so it's very likely it's going to run the race. So we can say that in the end, a vast majority of, remember, this is representing like natural selection, and we'll also look in real populations, so real like beetles or moths, that we can see this happen in. But the ones that are surviving, aka the ones that are winning the race, are going to be able to pass on their genes to the next offspring, and then those ones, when they run the race again, they're more likely to win as well. However, if you don't have the good mutations, you're not that likely to win, but you could still win. It's not like it's impossible, really, because you have shoes, you have like legs and stuff as well, and then you have water, but it's just going to be harder than those with the good mutations. This is why when you look in, let's say, a group of the same animals or same birds or same fish and stuff, those with the good mutations are going to be the most of the group. However, due to some like changes, or just like, again, mutations in the gene, there will be some people without the mutations that still are surviving for a little bit, and there's always going to be that little part of the population. So we pretend like it's a pie. We have the good mutations, like have a huge part of the pie. The like without good mutations probably have like maybe this part, and then if we look here, the really bad mutations, at this point, it's really impossible almost for you to win. So they would be very small or just almost gone, really. This is what it's saying here, since it's almost impossible to win. If you have all the bad mutations, almost no bad mutations will be passed on. This is possible, but hard to beat those with good mutations. Some no mutations will survive. So those are just, remember, the normal. However, the good mutations have the highest chance to really win every time almost. And so then that's why they're going to make up the majority. And so now I think we can settle for our first question here because we're doing good on time. And I just want to make sure we have enough time as well. So I think we'll answer one question.
All right. Our first question is that does mutations occur because the DNA just want to try out a new possibility and hope it will be a beneficial one, or is mutation just an accident?
So no, it's not really like that because the DNA does not have that kind of control. And as we'll see, because I'm going to explain some viruses as well in detail, you'll see really how this accident can happen. And I really feel like once you have a better understanding for viruses and how those mutations happen, you'll be able to see that for us, we're really covered mostly because we have a very low chance of mutations, but it is just on accident. And if we look back here, right, we look at this base pair change, this nucleotide change. Sometimes because okay, I'll do a short lesson here. This 3 nucleotide part, this is called a codon, C-O-D-O-N. But you just need to remember that it's like you're reading an alphabet or reading words. Each 3-letter segment is like one word. Each word will lead to one protein, but however, sometimes you change the base pair inside, it'll still do the same protein. So that's why that there's actually like multiple like codons, remember, are the words that will lead to the same protein, which is why sometimes you have the mutation, it doesn't change anything because the same protein's there, that's all that really matters. So that's why we can have some mutations that really don't do anything, they don't have any effect on us. However, we can have the chance as well if we have the mutations and it causes something bad to happen. That was a really great question, and I thank you guys for like paying attention as well to come up with that question. Okay, we're gonna move on here, and again, at the end we'll have more time, so make sure you write that question down. Here we're gonna go over a good example of natural selection. So we have these crows here, and this is pretend like a log. Now, just for the sake of this, pretend the orange ones are actually like tan, because that's what they're supposed to do in this picture, but it kind of got messed up a little bit. Here, we see that these are tan, so they blend in better with the trunk. However, these green ones, they really stand out. It's like you have some like blue beetle on like a completely white surface. It's gonna completely stand out. And this is why you see here the crows are just picking the green ones. So it's so easy. It's like you have like food right in front of you and you can see it. It's not even like hiding from you or anything. So once they're trying to pick up all of these green ones since they can see them and they can eat them. Over a generation, since like we said, when there are less of the green beetles here, there's less that can pass on the green beetle gene. And that's why we see here there's a lot of green beetles in the beginning. After a while, it's about even almost, and there's a lot less green beetles. That's because more and more are being eaten, so that to the point that like any green beetle that's really there is probably going to get eaten, and it can't reproduce and pass on its genes. However, the orange ones are surviving. They're fine. The crows can't see them, so they're fine. And so they're just passing and reproducing on their genes to their offsprings. And we see at the very end here, we have almost all here are orange, or the orange tan color, and the green are completely taken out. However, we will still have some green, but it will be very, very small. Again, this is just due to some changes in the DNA. Here we can talk, there's gonna be a future seminar on really alleles and like dominant versus recessive, which will also help your understanding of this. We're not gonna get into that too much right now though. But the interesting thing is the orange beetles will not always be the dominant group. In this case, they are because of the background, because of the environment. But let's look at a different case where things actually change. Here we have the peppered moss. This is a very used example, and it's a very good one to understand. Here we have a white tree trunk, these white moss here, right? That kind of blend in. They have some speckles on them, right? And they kind of blend in there, right? But then these black ones are the ones that are actually like standing out, right? And so this bird is just seeing the black ones, just eating it. Easy money, right? But then the Industrial Revolution, where like a bunch of coal was burned and then the soot comes out, it starts to blacken the tree. Now the tree is like all black. So here is white, and then the black moss would stand out. But now the tree trunk is black, so the white moss stand out. And the good thing is we still have some black moss, remember, because we said that due to just some mutations or just how the genes are passed on of dominant versus recessive, it will actually still keep some black moss, which will cause the entire moth population to really flip-flop. So instead of having the white moss be the predominant or just the dominant like group here of the moss, we actually have the black ones here being the most and being the most numerous. They're covering everywhere because they're perfectly camouflaged. And now instead, the white moss are being picked and taken out. So that's the very cool part, I think, is because not only will it be static, meaning it just stays and just have like this example here where the orange is just always going to be the dominant group, but here it can actually flip-flop based off of the environment. And that's why like when the environment changes, it can mean a lot and have a lot of impacts on the different types of species as well. Okay, we can answer another question here too.
Right, so another question we have here is that, is it a bad thing for bad mutations to win since they will pass on the mutation to the next generation?
So is it a bad thing for bad mutations to win?
Yeah.
Is that the question?
Yeah, that was the question.
Okay. So the thing is, like, yes, it is, but here's the thing. We go back to our race analogy, right? We go back here, the bad mutations have like very, very small, like almost 0% chance of winning, just because you have the good mutation, like these really like strong people that are going to run super fast, and those are going to win most of the time. They have the without good mutations, they're going to win whenever the good mutations don't win. So the bad mutations have the worst equipment, no water, no nothing, and then they're not going to be able to win at all. So that's why a lot of the times, bad mutations will actually not be that frequent, just because that, again, we saw here, let's say there was in this example, or well, let's say actually in this example here, like we start off here and then there's a really bad mutation that doesn't make it white or black, it makes it like purple. That's the most like bright color there is. And so there, just because it's purple and it's so bright, it's actually going to be attract the bird even more, and it's going to cause it to eat it, so it's not going to survive at all. But like we saw with sickle cell anemia in the last seminar video that we had last week, that actually having a mix of the sickle cell alleles, which again we'll talk about more in the future, and just having like half sickle cell and half normal will actually give you malaria, will make you kind of more immune to malaria, versus having both the sickle cells where it's very likely you're going to end up dying because you can't get enough oxygen and you have blood clots, versus having just normal, where you're probably going to get malaria if you're in that type of environment with a lot of mosquitoes that carry that disease. So sometimes, most of the times, the bad mutations will take care of themselves and they're not really going to win by any chance. But again, we can see this change go from this tree to this tree color. And so if for whatever reason the tree turned purple, then the purple mutation would be good. And it really just depends on the environment. But like I said, for the most of the part, the bad mutations are not going to be surviving, and we don't have to worry about them being the biggest group there. That was an amazing question. That was really good, because I was glad that like using that question, I can explain a lot more to everyone else who might have been curious as well. Great. So we're going to keep moving on here. Okay. And then at the end, remember, write down that question if you still have it. Now that we have a good understanding of natural selection, we can kind of dive deeper, literally, into how this connects with Darwin and his finches. I wanted to make sure you guys had some background information before we started talking about the Darwin and his finches. So now we have another question here. Why are islands important for natural selection, right? Like, again, we talked about some characteristics of islands, and why do you think they're actually important for natural selection? They are very relaxing places to live. They're isolated, or you can build your own house on an island. So let's see what you guys think here. Okay, just give everyone a couple more seconds here to answer. Yeah, I think we can close the polls right now. Yep. And again, everyone always picks the right answer. They are isolated. Great job. And so we look here, this is South America, this is map, and we can see the equator, this red line right here. And North America, where we are right now, is like up here more. And then of course we have the other side of the globe over here, but I focused in specifically on South America. You see these little dots of islands, these are Galapagos Islands. And here we have Ecuador, which is the mainland, if you will. This has the huge continent, has all the land connected. So what Charles Darwin was doing after looking many times at these finches, he realized that although they're different, they seem to have like these common characteristics as well. And he really saw that what happened was, let's say there was one original finch population, I don't know, just the original finch population. It must have flown over from like Ecuador or some of these countries here just on the land, flew over and then gone to the Galapagos Islands. And as I bring up a map of more detail about the Galapagos Islands, and I'm going to explain what that means and how we got these different populations, these different types of these different species of these finches. Here, this is a map of the Galapagos Islands. So let's say that like the mainland is over here, right? We first have the finch population go here, right? But the key part that we just answered, right? The islands are isolated from each other. So let's say one island ends up having like a really bad drought, or another one has a ton of water. That's going to change the living conditions for the finches, the birds, remember, on the island. And that's really important that these are all isolated, because what happens is the original population here. Let's say a couple birds just move over here, they branch out here, then we have a new population here. And then we keep repeating this until we have finches on all these tiny islands and the big islands and everything. But all the climates are slightly different. And not only the climate can vary slightly, but again, it's not going to be crazy different. It's not going to be like bone dry and then storming rain, because this island is very close together. Excuse me. They're very close together. But what's going to happen is also the food sources, though, because the food sources can be varied slightly, because again, they're isolated, and that's a very big part of it. Here we have all the different finches, remember, that came from this original finch here. And if you look at their beaks, this is what I was going to talk about. We see we have multiple small beaks here, and they get sort of larger. And not only do they get larger, but the shape changes. The literal shape of the beak also changes, not just getting larger. So if we look here, I'm going to try to explain to the best of my ability here about the food sources that these finches have. This very small beak is good for picking up the small seeds, kind of cracking them because they're kind of soft, and then get the food from inside the seed and then eat it. So that's how that finch, the warbler finch, I believe, will end up eating that seed. Here we have a bigger finch, bigger beak. So maybe just a little bit bigger seed, and maybe it's a little bit harder, a little bit softer. Here we have a bigger finch as well, but this one, you see, this is relatively flat. If you can follow my cursor, the beak is pretty flat like that, right? And you have the top part here, and then it'll like go like that. This is the first one we really see a curve here. So that curve really will end up, if we go to the opposite extreme here, this huge beak with really a really curved beak. But that will eventually lead to the harder seeds that are really like compact, dense, and hard. And that means that you need a harder beak that's curved, that has some like sharper parts to it to really crack that's really strong. Because imagine you have a beak that's like this, you have a seed that's like this big, right? But then like you have this tiny beak. There's no way you could ever eat it. And then if that's the only food source, this is where the natural selection comes in. If this is the only food source, if the really big seeds that are hard, this warbler finch is not going to be able to survive. It just cannot eat it. And that's the only food they really have. Maybe they find some other seeds, but the main source of food is those big ones, right? And then over time, these mutations, right, that we're talking about can eventually lead to there being a bigger beak here. And then that will allow it to crack these bigger nuts and then survive. And so I'm not going to really go through all of them, but you can see as they kind of get smaller or they get more curved, and they change slightly throughout this just period of going to these different islands. Here we talked about it, the birds coming from the Central/South America and going to these Galapagos Islands, flying here. And yeah, here are the islands again. I actually want to say one more really interesting thing. So ever since Darwin actually went to these islands and documented these finches, like he published his reports and everything and came up with this theory of natural selection. But the interesting thing is that people are still studying those finches today. They still want to know more. And there was a lot of unanswered questions that were not answered just because he wrote down some stuff and made some connections. There's still so much more that we can learn from these finches. So it's very interesting. And one of the cool things is that since, once again, they're all isolated, the food sources actually differ. And when there is a drought that may affect, let's say, majority of the islands, what will happen is the actual food will change. Because as there's a drought, the plants and stuff, they're like, some plants are gonna end up dying that really need the water. So now you're gonna have more hardy plants, and it's more likely that they're gonna have these harder seeds, right? Because if you just think about it just a little bit, right? We have these really like hardy, strong, like can survive drought type of plants. And then if they have seeds that are gonna eventually lead to a new plant, they want it to be very hard and durable because it may not rain for a while. And then once it does rain, it can be planted, and then it'll just like being covered with mud, and then it'll grow. But versus the smaller seeds, these ones are more fragile. So they're just relying on it being rainy and ready just to like grow easier. The whole point of this was that what's going to happen is this food source of big nuts versus the small nuts is going to change. Let's say the drought goes away. The big nuts, not going to happen anymore. So now we have the small nuts. But what's going to happen is, we talked about how the finches follow the food. And the ones with the bigger beaks, yes, they can technically eat the smaller and less large nuts that are easier to crack. But the thing is, they are actually larger and bigger probably, and it's not as efficient for them. Because it's like, I think it's sort of like you take like a backhoe, the machine that has like the big scoop at the end, and you use to dig holes and you use it for construction. If you take that and you want to dig like a 1 foot deep hole, it's just like, it's the amount of energy and money you have to spend to get the entire thing there to dig it is just way too much, and it's like overkill really. Whereas you have a shovel, it takes a small amount of energy, but it's really like no money. And then that's the same idea here. The small birds, they're going to be more quick, and then they can get more easier, where the big birds, they're going to have the bigger beaks that are harder. And in reality, even though like you would think, oh, I think they can actually still have the small seeds, maybe just because how their beak is designed is not meant for that. So then they're not going to end up surviving. Yeah, now we can answer another question as well.
All right, so another question is that, does everyone have mutations?
Yes, everyone does have mutations, because we already talked about like how the mutations are just in our DNA. So even different species will have mutations. And I'm definitely going to end up doing probably a seminar on gene expression, which is how we get from our DNA to the actual traits, because I know people have been very interested about that. But that's also the key part, because we have in some spots, we have this mutation that causes these animals to be albino, which means that their hair is white, their eyebrows, everything, all the hair, their skin is super white and everything. And it's to the point, it's so pale that if they live in places, let's say like just think of Hawaii. Think about how much sun is like everywhere, right? Or like around in the rainforest, there's a lot of sun. And if you have this really pale skin, what'll actually happen is you can end up dying, because your skin is just not really made for it, made for that amount of sun. However, there is a difference though, because people that have lighter skin shades, like now that have that, they're fine, because they have like lighter skin shades. But compared to someone with this albinism, is what it's called, they have super pale, like it's really, really pale. And they actually have to like wear long sleeve shirts and put on like huge hats and stuff when they go outside, because their skin does not, their skin does not have this thing called melanin, which is responsible for the coloring of our skin. So people that have darker skin will have more melanin. So what happens is this melanin is like very little or barely existent in these people with albinisms. And yeah, so that was a great question. Actually, I think we can take one more too.
All right, yay. So another question is, do the finches consciously adapt to their living conditions?
That is a great question that everyone I know wants to know. And like that's one of the biggest misconceptions about natural selection. It's just like the giraffe. You think about the giraffe, most people are like, yeah, it was maybe just a horse, right? And the horse was like, I see some fruit at the very top. Maybe just like strain my neck just a little bit. And then people like, oh yeah, each generation they do that, and they have like a little bit longer neck and a little bit longer neck and a little bit longer neck. But we see that with these mutations, it'll actually, like in the giraffe example, we have a mutation for a slightly longer neck. Those ones are going to be able to reach and longer legs as well, because the giraffe has really long legs that help. But it's going to be able to reach some fruit that the other ones cannot reach. And over time, remember we said the ones without the beneficial mutations, they're not going to be prevalent anymore, really. They're not going to be able to survive if you can't get that food that this other person can get super easily, right? So over time, what happens is, as the mutation just over time will happen occasionally in copying the DNA, causing there to be a longer neck, and then eventually it will reach like the tops, and then that's how we get the drops. So we're gonna move on here a little bit, just in case. I want to make sure I don't want to have it be like we ask a question now and then like I answer it later on. So I want to get through some of the material first too. So here we're going to talk about viruses. Here, as everyone knows, is COVID-19. This is the actual virus. And we see these different proteins on it. So yeah. Now with viruses, actually, we'll stay on this slide here. Viruses are very interesting because they have this really, really big and prevalent, and they're a good example of natural selection. Because everyone knows how we have to have new vaccines, not even for coronavirus, but just in general for the flu, we have to have new vaccines every year. And I don't know about you guys, but I've always wondered, why is that, right? Why don't we just have one vaccine that just cures for everything? What actually happens is with the flu and many different types of viruses, their structure is very different from ours on the inside. And so what happens is they're they don't even really use DNA. And so what they do is, you can think of it like hijacking. So what happens is the virus won't really be able to do much by itself until it attaches on or gets in our body. Then it can use our cells to make more of the virus. And then what it does, we can talk about this too later, but anyway, it has this short genetic information, but it's RNA that we talked about, which is very interesting if you think about it, because we use RNA in our bodies, but it's for the proteins only. But these viruses, instead of having DNA, they have RNA. They'll actually go try to pass through and then get to the cells. Then they will insert their RNA inside, and then it will try to go all the way into the nucleus, and basically it'll take control of the nucleus and just make more viruses instead of doing its normal job. And a very interesting thing is, if you think about all these symptoms of having a flu or being sick or the common cold, you have runny nose, you have maybe just runny nose, you're sneezing a lot, you're coughing, stuff like that. And if you think about it, it's very, very interesting, because what your body is doing in response to the virus is it's creating more of the your snot and stuff in your nose to come out, to flush out. If any virus can go in through your nose, it wants to flush that out, right? That's also why you sneeze, because most people know you can sneeze at 100 miles an hour, right? So when you sneeze, all the air in your lungs is really coming out in a very fast instant. So if you breathe in any virus, when you sneeze or you cough really hard, it's going to come out. And so your body is trying to expel as much of the virus, because when you have more of these, they're going to be taking over the cell more and more, and it's going to cause more and more problems. So here I'm going to talk about the natural selection part too. This is another virus called HIV. It's a very, very big part of study right now because it will actually degrade your immune system. And so it does like really bad things with that. So that's why people are really researching on how to develop a vaccine. But one of the reasons it's so difficult, and also with this coronavirus, because if you think about it, many people are like, well, why didn't they just have a vaccine ready as soon as they realized it was coming out? Well, it's not that simple. Because what ends up happening is we have the RNA, right? When when the virus hijacks the cell and puts in the RNA, what ends up happening is that it's not really using all of the machinery that was used to check and double check that the viruses have the correct like base pairs and RNA and there's no mistakes. It doesn't do that. It's just worried about getting out as fast as it can. So this is where we really see large amounts of mutations. And like we're talking like, let's say we have a virus that's dividing, like it can have random mutations that just end up happening because there's so many mistakes. Yes, some may die if it's bad, but there's just overall such a large number and so many like mistakes that are happening that it'll cause completely new viruses in a short amount of time if they keep dividing. So that's why when we make new vaccines every year, we have to then target a new type of like the virus. And that will be probably the most prevalent. So the virus that is infecting the most people and everything. So then they're gonna make it mostly for that. And then the next year the virus will change and then they have to make a new one for that as well. So that's why it's very hard because these ones can be like very genetically different. And this is why when in our cells, as I'm going to show in the next slide as well, we have our own like immunity response and it will recognize it based on the proteins on the outside. So we see here, we have this like virus here, the small mutation can change the protein shape, or it can change like just the protein, like what it's made out of. And what our cell does is kind of like just seeing and like let's say like reading a sign on a car, right? And you're at a border and you're like, oh, okay, well everyone with like let's say X cannot pass, like they have an X sign on their window, they can't pass. But then you start seeing people with Y window, like Y's on their windows, and you're like, well, I didn't say no. So then the immune system doesn't recognize that as a virus really, and then it can actually get past for a while until eventually it'll be recognized. But the vaccines, what they do is they will take this virus, they'll kill it with like heat and like a bunch of stuff like that. But then, so they have the dead like shell, if you will, that has the proteins. Then it'll be like kind of injected into us, and then our immune system will be like, that is a virus, and then it'll start like trying to have the immune system respond to it and make these things called antibodies, which help fight off the viruses and kill them. It'll make them. So then what happens is, let's say you go outside and then your friend has the flu and then you are like next to him and you're like hanging out. But if you already have the vaccine, your body already has the antibodies. So when you breathe in, maybe let's say that the virus or flu that your friend has, you're not going to get sick because you are ready with like all your cells are ready. They're ready to fight this like flu and everything. And so they're already prepared. Whereas you just catch it without the vaccine, you're not prepared. Your body's not prepared and you're not ready. So then you have to go through the whole thing of being sick. And that's very dangerous, especially for like the people that have like HIV in this instance, because their immune system's not that good. Or just if you have medical conditions, then it becomes very hard for you. So yeah. So we're going to move on now to how this applies to human evolution. So they've done like studies here, and they've shown that like the chimpanzees, based off the DNA. So what they did is they took all the DNA of chimpanzees and all the DNA of humans. They used this like this process of kind of analyzing them and like writing down the base pairs. So then what they did is they compared them. They found that it's actually 99% like the same. But I'm going to talk a little bit about why it's actually different and like why we obviously look so different and act so different. So, oh, well, I think I already gave this away, but we're going to ask this anyway. How did scientists determine who our closest relatives were? Yeah, it was very interesting because in the very beginning, before they had this answer, but before they had it, they would actually like try to compare and see based off of like just the appearances, like if they were actually the closest relatives or not. But then we found out that that's not really the accurate way, because we can look very similar to something else yet not be related at all. So that's why you really need to do the like kind of biochemistry stuff behind it, and that will really have concrete evidence for it. Okay, we'll give everyone a couple more seconds here to vote. Okay, I think we can end the polling now. Great job. Clapping for everyone. DNA was the correct answer. Okay, and so we're gonna move on here. Yes, DNA. We share 99% of our DNA with chimps, like I said, and I'm gonna talk about in the last couple slides here of the seminar, like why we're still so different from chimps. But here I have a video kind of talking about like the human evolution. It's kind of long, so I'm gonna try to like speed it up just a little bit, and this time you guys will be able to hear the music, so it should be pretty good. Then after this, then I'll explain a little bit more, and then we'll go into the questions from last seminar that we couldn't answer. Whoops. Here we go. Right. Let me speed it up just a little bit. So they're talking about the changes from the apes here, and then they're really highlighting which changes they showed that suggest the like upright walking or differences. I think this video is just good to have like a background, this background information in general. Yeah, I think it's a really good video because it kind of shows where they found these fossils and like where they believe they were from. And they're talking about how over time, like, so some of these mutations in natural selection kind of led to these advantages that eventually caused, yeah, as you can see, caused them to eventually survive, and then only one species be the dominant species here. And yeah, so then they're talking about how cooking, like using controlled fire, will actually increase the calories that you can get. And at that time, it was very hard to like try to survive off just the like raw, like I guess you could say vegetables, because like there wasn't much calories in it. Yeah. So then like when you cook something, like let's say potatoes versus like baked potatoes, the baked potatoes are going to end up having a huge, way larger amount of calories. That's why like when you eat French fries, it's like a lot of like calories as well. But this helps them because now they don't have to really worry too much about starvation, and then they can continue to like advance. And then now we go back to that overall one here. And yeah. So I hope that helped you guys. And now we're going to talk about a very interesting person here, is Allan Wilson. Here he taught at like UC Berkeley and also worked at UC Berkeley for a while, but he was actually originally from like New Zealand. And we'll talk about what the great things he did, because he's actually not that well known to most people as, let's say, like Charles Darwin, because he's like Charles Darwin's like the father of like evolution. Everyone knows him really, but this person is not as well known, but he did just as important things really with connecting, like is more recent in the more recent years, but connecting like the biochemistry, which like we talked about DNA, for example, all the molecules and the like the structure. We said biochemical structure, remember? He's doing really that, but he's connecting with evolution as well, which is something that we're going to talk about here, but not many people actually made that connection back then, and it led to really some big discoveries as well. Here, he grew up in New Zealand on a dairy farm, and then he went to King's College. So he ended up staying and working at UC Berkeley for most of his career, but he was very interested because he realized that most people in this field are either an evolution person or they're studying like chemistry and biochemistry. There's never like any interlap. So he really wanted to make this unprecedented connection between biochemistry and evolution and bring them together to really make this higher like level of understanding that we can have by combining different parts together. And yeah, most scientists were either one or the other, and he faced some opposition to this because they were thinking like, well, he's not even focusing on our field. Like, let's say that evolution people, they're like, well, he's just studying, you know, biochemistry and stuff. And the biochemistry people were like, why is he studying evolution? So it was a very like relatively new thing to do, and because these two fields, to most people, they seem so different. And he really pointed out how most evolution people did not know that much biochemistry and vice versa. And he really felt like this was a big problem, and how he had this like great vision for how he could connect the two to end up getting even more support and just everything else as well. So here we have the idea of evolution and biochemistry. And here he worked and taught at UC Berkeley, also was like a researcher as well, right? And he had a very interesting passion here for birds as well. And he just liked to study birds as well. So that was very interesting. So what he actually did was very, very like groundbreaking stuff as well. So if we go back all the way, let's say to here, we have this like kind of part where we're all diverging and we're creating like these different species, right? And then eventually it's just us. Well, let's go here. Yeah. So he compared the proteins in these diverging species tissue. And he said that the differences between these like proteins and the DNA will show how far away they are from the ancestors. So we said like the chimpanzees, only like 1% is different, or like less than 1%, right? So that really shows how close we are to them, and like DNA wise, our DNA is very similar. However, he realized there's actually a great difference between us and chimps. So I'm going to get into that a little bit, but like it's mostly the DNA matches up, but there's a different part of it too. So, but he wanted to prove that the really DNA matches up, because when you can prove that the DNA matches, then you can say that like, yes, on a like molecular level, we're sharing the same DNA as well. What he did is, you can see here, he used blood samples from the tissues. So it'd be like a human or like a chimpanzee or other stuff like that. And they also included stuff like dogs, just to see like if there was any connection. And he applied these antibodies to the blood to find the correlation, to see how closely related they are. Now you might be like, we talked about antibodies, but like they just like protect us from viruses. How is that going to work? But I'm going to explain it here. So he took the antibodies from humans, and then he used like rabbit cells to actually generate more of these antibodies. And that's kind of the part where we go with like the gene editing, and then we can actually have animals be producing these like special like enzymes and stuff like that. Like there's these goats that are being used to produce like life-saving certain like enzymes and proteins that are needed for people, whereas it'd be really impossible to get them. You would have to get them from a human in order to give it and save somebody's life. So that's why they've changed some of the just the what they've done is, for the goat example, they did it so that the enzyme is secreted in the goat's milk, and then so when people drink the goat's milk, only that specific goat milk obviously, but it will help those that need the specific enzymes that they, for whatever reason, like the mutation, they don't have right now. And so yeah, so then using the rabbit, they generated more antibodies. And then what he did is, since the antibodies would be actually in humans would be able to compare with just the structure of these other like the cells and the other antibodies of the closer other species. So I'm going to explain like what he did. I don't have a picture for this one, but he would take the blood, put in like little vials, and he put them in like a kind of just like table sort of thing. And then so what he would do is he would add the antibodies, and we talked about hemoglobin. So hemoglobin is like the part of our blood cells that carries the oxygen. So using these antibodies, the blood will actually change color, and the brightest color obviously would be ours, which is human. But then the next brightest color was like chimpanzee, and then they used many other monkeys as well, and they saw that it became less and less bright, and also became like more clear. And the most clear would show that that's the most farthest away from us in relation to us. And let me see the next slide here. Oh yeah, so this is when I go on the questions, but let me go back here real quick. So what I was saying is, like, we share 99% of our chimp DNA, or of our DNA, excuse me, with chimps, but why do we look so different? Why do we act so different? How do we have this like higher level of intelligence rather than like they do, even though they're very smart animals? What is actually happening is, think about DNA as a book, right? We have this huge textbook, all the DNA, right? And we let's say we share, right, 99% of our DNA with the chimps. So 99% of this really big textbook is with the chimps. And let's think you're a student, right? And if you read that book, it depends on what pages you read. Because you can have all that DNA, but if you don't read any of it, then nothing's going to happen. You're not going to learn anything, right, in the textbook. But if you read the whole thing, then you'll learn a lot, versus you read half, you learn half, etc., right? So that's the same thing that happens here, because the DNA, even though it's almost identical, the parts that are expressed and the parts that are actually going to end up coding, because we talked a little coding for like proteins, which will make us up, which I'm going to talk about more in gene expression. But what ends up happening is that these will code for like the proteins that make us up, right? But then yeah, so then what's going to happen is there's this thing called introns and exons. You just need to know that exons are the ones that stay together. So let's say, actually, this is kind of hard to explain here. I'm going to use a whiteboard so everyone can see what I'm doing. Okay, let's use blue since our favorite color here. We have our DNA strand, right? And we want to like replicate it, but there's actually a large portion of DNA that doesn't really code for anything. So let's say we have these red parts here, and then I'll change it to green, right? And then we have like some more red maybe. Okay, and let's say, okay, let me just add one more green here. Yeah, let's say that only the red will actually code for anything, right? So what's going to happen is we're going to have this like special like kind of protein like called a spliceosome, right? It just comes in. It's going to, actually, it's very interesting what it does. So it's going to actually like, it'll cut this out. I'm trying to draw an arrow there, so if anyone's confused. It'll cut this portion out, remember, because this one is the one not coding for anything. And it'll also go here, do the same thing, cut it out. And we can talk about this later too, but the really cool thing is, if you're wondering like how does it cut it out, it'll actually form this thing called like a lasso, or in science a lariat. You don't really need to know that, but it'll form this sort of loop thing here, right? And let me draw the red, and then this will be like that. So now it can kind of pinch together, and then we draw the spliceosome here that's kind of like grabbing it, or actually, sorry, the spliceosomes will go inside here, and then they will kind of form this loop. And then this will lead, the mouse drawing is kind of hard here. Okay, this will lead to eventually the green part that we didn't want, right? Is going to be taken out here. And then we have the spliceosomes again ready for use. So this is how this whole entire process will go down to here. But let's say, for example, we switched it. Let's say instead of taking out the green, we took out the red. Well, what's going to happen is we're going to have different DNA that ends up like being expressed. So that is why we can have the same DNA, but how we express it will change what we look like and how we function and what we do, which is why we could share 100% of our DNA with another organism, but if we use different parts of that DNA and we don't use all of it, because no real like organism isn't going to use all of their DNA at the same time, and if we use different segments, we're going to end up becoming different things. So that's why the kind of explaining that is like why we're very different, even though let me close this here. Even though we are, I need to share my screen again. Even though we are the DNA is like almost completely the same. So that's how we can account for the differences, like the physical differences here. Okay, now we're gonna go over some questions people had from last seminar. How is DNA different from RNA? Well, we talked about the double helix. Oh, just a side note, so for everyone else, that was like all the evolution stuff we're gonna be covering. So now we're gonna be going over like these kind of questions, and then we're also gonna be going over the evolution questions people have. So if you guys have some like other conflict, just you guys can go, and then you guys can watch the recording later on YouTube. Here we have the double helix, and here we have the single helix. And here we have this different sugar here. This is called deoxyribose. It's called ribose. Literally, the one difference is right here. You see my cursor? This is a hydrogen atom. This is an oxygen and a hydrogen atom. That is literally the only difference between the sugars. Another part is we talked about how adenine and thymine and guanine and cytosine are the 4 base pairs of DNA. However, this changes in RNA. Thymine, there's no thymine. It just becomes uracil, this different base pair right here. And so instead of adenine going with thymine, thymine doesn't exist anymore. So then it goes with uracil. Guanine and cytosine, however, keep going together. But that's really just the difference, I feel like, between these two. And I really hope I cleared up everyone's questions, because this was a very interesting and a very major question I felt that we didn't have time for last seminar, which is why I want to cover it now. Also, another big question, DNA curls. So DNA curls is really the variations in DNA. This is actually pretty high level, so I was kind of glad that people asked about it here. But we have 3 different types of DNA. We have the A DNA, which is right here. We have the B DNA, and we have the Z DNA. So you can kind of tell that the B DNA is the stereotypical normal DNA. And it's really just the normal DNA that we use in our body. But A DNA is slightly different. As you can see how this turn works here, and this helix is formed. This one's like closer, and it's more, you think about DNA like a spring. A DNA is more compressed. B DNA is probably like normal. And then Z DNA is like the spring becomes like just like a rope or something. It's very just like unstable. So so aDNA here is like a mix. We can talk about this too later, but DNA and RNA can actually sometimes mix a little bit, or the double-stranded RNA, because we said how RNA is single-stranded. Sometimes those will join up and form a double strand. But the bDNA, normal DNA. However, zDNA, I searched up a little bit, researched some, and it actually leads to brain disorders and other diseases because this structure is so unstable. As you can see already, it's like already flopping, and this is just like a stationary image. And we look at this is the molecules. You can see it's all over the place. This stuff's not like uniform connected, and this is looking from the top down. So if you were like look at like the DNA from the top down, this is what you're seeing here. And we see how the variation is on how they curl and how they like turn, and then the actual structure. And for the actual DNA curls, these two here, I put R's for right. So they're right-handed curls, and this one's a left-handed. What that means is, this might be like flipped on your side, but just take your right hand and then just curl it in naturally like that. That is what a right-handed curl is like. And then it'll keep rotating like this. So that's the same as like counterclockwise. Left-handed, take your left hand, turn it in like this normally. That will have it go clockwise. So these variations in these curls can lead to different functions, which is why ZDNA, the only left-hand curl, and very unstable. So yeah. Also, someone asked a very cool question is, can we make artificial copies of DNA? Great question. Polymerase chain reaction, PCR, is how, this is the main way we do it. I wanted to kind of simplify it because there are a lot of different ways out there, and there's more being tested more and more, because everyone is looking for a faster way to copy DNA so they can study it more and even easier. So here we have the original DNA. We split it up because what happens is it's denatured. That just means that we heat it up, all the bonds just like kind of break and it separates. So whenever you heat up something, this is why your body temperature really can't get up that high, because otherwise you wouldn't be alive anymore. But if you heat something really high, everything's going to denature, and the proteins and everything inside will just kind of fall apart because it's breaking the bonds. Like on the molecular level, is actually breaking the bond. So we said how there's like the guanine and cytosine that have these hydrogen bonds, right? It'll break those bonds, which is why we see the DNA splitting. Then we talked about this in our last seminar here, but if you forgot it, you missed it for some reason, go check out our YouTube channel. We talked about a primer that we put down, and then we begin to go just like, we have these nucleotides in the solution, because this is happening inside of like a test tube. It's not inside the actual cell anymore. We have the nucleotides in a solution, then it's dragging them and putting them in place. Then we repeat the process. We break it again, add the primers, keep replicating, break it, keep going, break it, keep going. And this way we can create huge amounts of artificial DNA. So yeah. And the last one here is, how was DNA discovered? This is a big question. I know that a lot of people had. It was very interesting, and I'm really glad you guys asked it because it's a really important question. Here we have these 4 people. There's a couple more, but like these were the main people here. We have Linus Pauling, who realized the proteins have spiral structure for, this is all DNA again. We have Francis Crick and James Watson and Rosalind Franklin. There was also this scientist named Chargaff. He came up with these special ratios. Remember, because we go back, to these 4 bases here, the thymine, the adenine, the, I can't really read that, but like guanine and cytosine, right? And he realized, he actually took the ratios, like the percent, if you will, of how many there were of each. And he realized that the adenine and thymine, the amount was literally almost the exact same. And the difference, the reason it wasn't the exact same is because back then, like the like technology and equipment they were using was not like that accurate. But today we have actually proven that is the exact same ratio with better technology and equipment. And he also noticed, so we said adenine and thymine, the amount is the same. Guanine and cytosine, the amount is also the same. So he didn't really know what to do really with that, but he just found another discovery about DNA. However, that was a huge part here. So that was Chargaff, remember? And here we have Linus Pauling, kind of realized the proteins have this spiral structure. So it's kind of like you have all these pieces of information from different scientists all coming together. And the people that really discovered this was the James Watson, Francis Crick, but also Rosalind Franklin as well. Because James Watson and Francis Crick, they kind of pulled these information together, and they also looked at this picture right here, which was from X-ray crystallography, which I'm going to explain in a second, that Rosalind Franklin actually made. So they also took a little bit from her and then used their like thinking, they put on their thinking caps, they put it all together, and then they came up with this model here that they actually built of the DNA. So it's pretty cool. X-ray crystallography is what was the main way that we looked at DNA at that time. And what you have is you have the DNA arranged in like a crystal. So then you have it like side by side. Then you take this X-ray tube, it shoots like X-rays, just like X-ray beams. You have this little slot, so it only goes through, and then it'll be focused more, hits the crystal. Then you have this kind of shield in the center to prevent it from also going to the center. And then what happens is the X-rays are going to go through it, but then they'll spread out. And just like how we take a picture sort of, or we can look at something on like the wall, if we have a flashlight, the light will go onto the wall and we can see it. Just like that, the X-rays would go from the tube here and they would go onto the wall, and then they would be captured by this special plate. And that's how you have your photo, really. And we look here, it's kind of obvious to us, but at the time it was still kind of debated here. We can almost kind of see that we have this like this double helix structure here, right? Because this is like the middle, you can see middle segment and like that. And sadly, Rosalind Franklin died of cancer before she could actually get the Nobel Prize for helping discover this. But James Watson, Francis Crick, and also I believe, James Watson and Francis Crick, they received the Nobel Prize for discovering the structure of DNA. So, and actually, so very interesting and very glad you guys asked that question. Okay, so now I know everyone's been dying for this moment, but now I would like to thank everyone for coming to the seminar, and if you guys have any questions, I'm going to answer them.
All right, so we got a lot of questions right here in the chat section, so thank you, everyone.
Okay, wait, before we start real quick, just if you guys have questions that we didn't get to, feel free to like either send them to like the host or send them to our email, or use this, you can use this here, this code here, and then scan it and then send it to us, because in the future at the next lecture I would love to answer everyone's questions, so I want to make sure I can add them just like I did this one and add them at the end.
Yep, that's right. And so our first question is, will every animal adapt to their environment?
That's a great question. Great question. Very, very good thinking. So yes and no. Some animals will, not all will though. Some will and some won't. There are certain factors that will actually cause them to be more likely to like adapt quicker than others. Like we see these viruses that can adapt super fast, right? Because they have so many mutations. But animals, on the other hand, they don't have as many mutations like that. Like the viruses have insane amount of just errors. Like it's crazy. But like us animals, thankfully, we don't have that many errors like that. But let's say we have like a group of, let's just say like 100,000 zebras, right? Versus a group of like 100 zebras, right? And they both start off living in the normal conditions, but let's say like for some reason something changes, right? And then they're needed to adapt to that situation way better. Let's say the water becomes even more scarce or something like that. What's going to happen is in the population at the very beginning, a lot of animals are just going to end up dying, sadly, because just they're not adapted. But as there are more like offspring in the larger group of zebras, there's more of a chance that there will be a mutation. And then that mutation could be beneficial, like we talked about, that could lead to them being able to adapt better. However, we only have in the other group 100 zebras. 100 zebras like nothing. And that over time, right, you have, let's say, like no more water, or like the food's very scarce. That's going to already kill easily, like sadly, 100 zebras. So they're not going to have the chance because it's too sudden for them. It would be really helpful if it was gradual, very, very gradual, and it gives them plenty of time, because like we said, it's not voluntary. They can't control themselves. They can't be like, okay, I don't need any more water now. Like I'm perfect like that. But it's just it'll give more time for the mutations to happen, and eventually those could be beneficial, which is why when you have a larger group, it's better. Whereas we have these in the news and everything, we have these like endangered species. And the reason everyone's so worried is because there's literally like 2 left on the earth. And if something happens to their environment, they can't adapt because there's literally only 2. And then if one of them dies, then it's really over. So that's a very sad part there. But that was a really, really great question.
All right. Before we get to any more questions in the chat here, I see Richard with your hand up right now. And you may unmute yourself if you have a question to ask. You have a question?
Yeah, I just like want to ask like how many species of finches there are actually in the world, not on like just the islands.
Oh, okay. Well, like we looked back there and then there was, okay, the thing is like a finch, there's going to be a lot of finches. I would say probably like at least like in the hundreds of like species. I don't know exactly off the top of my head, but the finches are not just limited to those islands, because the finch is a very kind of common type of bird as well in other parts of the world. And in other parts of the world, there's going to be different species. So I would say it's going to be a pretty big number because we're talking about the whole world, where there's a lot of different bird species and stuff like that.
All right, that was a great question. Thank you. So our next question is, how often exactly do mutations happen to us humans?
Great question. So it's kind of hard to say because we have to look at the entire human population, right? Which is like 7 billion people. And we kind of have to look at like how often like we're reproducing and stuff like that. But we can say that it's pretty rare because a vast majority of the population does not have the mutations. And like we said, it's more likely that a mutation is going to be bad than it's going to be good. So majority mutations are going to be bad or they don't do anything. So that's why there's many different disorders where there's mutations. Like we said, sickle cell anemia, where if you have that mutation, if you have the full mutation, which again we'll talk about when we go over like inheritance and stuff, when you have that full gene, the full mutation, you're like, there's no way you're going to really survive. So then you're not going to pass that on, you're not going to spread it. And it's also very rare because let's say you ask your friends, you ask everyone in your school, do you have sickle cell anemia? No. Like if you have the full blown thing, it's pretty rare. So that shows us that really this mutation, like this happening and there being a bad mutation or a good mutation, is also very rare. Because otherwise there would be like, it would be very normal for us to have different things happening with our bodies like normally, which is not really what happens.
All right, that was a great answer. Our next question is, why do only some of the chimps evolve, but not all of them? What determines which chimps evolve?
Yeah, so like we looked at the finches, right? We said that the common ground finch was the original species that flew over from the South America. So we saw that these different islands had different, slightly different climates, slightly different resources and food. This kind of caused it to there be like a specific species kind of tailored, if you will, so specifically for that island. So just like how in the world there's so much land, so much space, we have like deserts, we have rainforests, we have forests, we have like oceans, we have all these different types of like biomes, which are like, just different areas where animals can live. So this is why we have different chimps that are going to end up evolving, and different just monkeys as well, because as they move and migrate, let's say to find more resources, they encounter a new habitat where they're going to have to adapt to that habitat if they want to survive.
So it's very late right now and we can get to one more question. So our last question is, since we share 99% of our DNA, can chimps get the coronavirus?
So the thing is with the viruses, there's actually like a big, kind of like major study that's happening where the transmission of animal viruses to humans and human viruses to animals is actually kind of a big deal, because as we saw, like some parts of like the Ebola outbreak where came from a lot of these like bats that presumably had it, and then people ended up eating them, and that's how it kind of spread like that. So it can happen, and the thing is, like, for some animals, they're just different. And I have not been studying like chimps my entire life, so I don't know like a lot about the details of how exactly they work, because again, many animals have different adaptations that separate them from us, and different, again, gene expression of their DNA that separates them from us. So some viruses, again, we can think of it like as us as well, but some viruses will not necessarily affect them as they do us, but some will. So for example, let's say that your friend is not immune to the flu, but you are, and then you just have some flu like particles on you, and then your friend gets the flu, he's gonna be very sick, right? Just like how the bats can like transfer like they could have transferred the Ebola, and then that's how the people ate it and they got very sick, right? But if you're immune to it, or there's a different part, it's like you're this thing called a vector, which means you can transmit it, but it doesn't affect you. So one example was the bats, right? Because the bats were not really dying; they're just normal bats, but they carried it, like, let's say, in their fur and stuff like that, and they were kind of either immune to it or just didn't affect them. Same with mosquitoes. Malaria is also like a blood-borne illness, and the mosquitoes themselves do not suffer from malaria, but they can transfer it using their, like, they have this really, really tiny little like kind of beak sort of thing that will like puncture in, and then they'll get the blood, whatever. But they can actually transfer it because they're actually is not going to affect them. So that's the answer. That was a good, really, really great question. I'm glad you asked it.
All right, so that's it for today. And thank you so much, Ryan, for this awesome seminar.
Yeah.
You really made biology super fun for all of us to learn. And the seminar was particularly long, so thank you, Ryan, for being so thorough and informative. And thank you to everyone else for paying attention for the entire time.
Yeah.
I got so many questions here in the chat, so big thank you for everyone who asked questions and actively engaged in our seminar. And again, thank you for joining us today and sticking to the very end. And also make sure to follow us on our social media accounts, where we will be posting updates for seminars. And make sure that you subscribe to our YouTube channel, where we will be posting our seminar recordings. And we will see you next week for more seminars. All right, so thank you so much, guys.
Thank you, guys.